Imagine for a second sitting in one of those really sterile, fluorescent-lit consultation rooms.
Oh, yeah, the ones that just smell like rubbing alcohol and anxiety.
Exactly. So you're there, the doctor walks in, looks down at a clipboard, and delivers a diagnosis that just makes the floor completely drop out from under you.
Right.
You have cancer,
Mm-hmm.
and, I mean, the room starts spinning.
Yeah.
But then comes the real gut punch.
Yeah, the part they don't show in the movies.
Exactly. Because when you actually ask how they're going to cure you, you realize that the absolute best modern science can offer is, well, it's essentially a highly educated guess.
Yeah. I guess based on an outdated historical average of people who, frankly, aren't you.
Right. You're an individual. You have this entirely unique biology, but your medicine is about to treat you like a statistical average.
It's a profoundly disorienting moment for a patient. I mean, you walk into a hospital today expecting the absolute precision of 21st century personalized medicine.
You want a silver bullet.
Right. You expect them to look at your specific tumor, figure out its exact weakness, and neutralize it. But the underlying reality is that you are met with what is basically 20th century statistical probability.
Which is terrifying.
It is, because the system doesn't actually know what will cure you. It only knows what has statistically delayed disease progression in like a cohort of 1,000 strangers who just happen to share a few superficial biological markers with you.
And that right there brings us to the core mission of our deep dive today. We are unpacking this completely viral, paradigm-shifting manifesto. It was posted on March 21st, 2026 by a biologist named Parmita Mishra.
Yeah, and the place it was posted is just as fascinating as the content itself.
Oh, totally. It's hosted on this deeply fascinating futurist blog run by Paramendra Kumar Bhagat. And we're going to explore what Mishra is proposing here, which is, honestly, a radical new blueprint for curing cancer.
She's advocating for nothing less than moving the entire discipline biology from what she calls forensics to film.
Forensics to film. I love that phrasing.
It's brilliant. That distinction, the shift from forensic snapshots of, dead tissue to a continuous live-action film of living cellular behavior that is the absolute linchpin of everything she's doing.
Right.
It forces a complete reframing of how we approach drug discovery, how we do clinical trials, and specifically the integration of artificial intelligence into biological sciences.
Well, and before we really get into the weeds of the science, I want to set the scene with the source material itself, because the vessel for this information, it's just incredibly unique.
Yeah, we aren't exactly reading a peer-reviewed paper isolated behind a massive paywall in Nature or The Lancet here.
No, not at all.
Hmm.
This is a raw, urgent thread captured on a blog that aggressively juxtaposes these highly technical biotech breakthroughs with like really heavy macroeconomic predictions, geopolitical tensions, and this emerging philosophy called calciism.
It's quite the mix.
It really is. You have the absolute bleeding edge of scientific salvation sitting right next to the chaotic reality of global political upheaval.
And honestly, it serves as a perfect time capsule of the technological zeitgeist of early 2026.
Oh, absolutely.
It's a moment characterized by this intense simultaneous convergence. Artificial intelligence, microrobotics, synthetic biology, they're all maturing at exactly the same time.
Right, and they're just crashing into legacy economic and healthcare systems that simply were not built to handle them.
Exactly. And what makes Mishra's contribution to this era so compelling is that she is introducing what she calls an operating system for biology.
Which is such a tech-heavy term.
It is. And it's so incredibly rare to find a source that perfectly bridges this raw, heartbreaking emotional motivation with, like, hard-nosed, relentless engineering.
Right. But to understand why her solution is so necessary, we first have to understand the specific mechanics of what she's rebelling against.
We need to look at the current standard of care.
Exactly. Like what actually happens in that consultation room when the doctor initiates the current protocol. Let's deconstruct that modern oncology machine a bit.
Yeah, let's do it. So, on the surface, the protocol sounds like a total marvel of modern science. When someone receives that terrifying diagnosis, the immediate next step in advanced care is usually genomic sequencing.
Which sounds incredibly high-tech.
It does. A surgeon takes a biopsy of the tumor, that tissue is sent to a specialized lab, the DNA is extracted, and a massive sequencing machine reads the billions of base pairs to identify the specific genetic mutations driving the cancer.
Right, so the doctor tells you, we are sequencing your genome. It really gives the patient this illusion of absolute precision.
But the illusion of precision is exactly the problem here. First off, consider the timeline. The patient doesn't get those results in an afternoon.
Oh, right.
Extracting, preparing, sequencing, and analyzing that DNA takes time. The patient typically waits 2 to 3 weeks for that genomic report to actually come back.
2 to 3 weeks. I mean, when you have an aggressive cancer, a 3-week wait is an absolute eternity.
It's agonizing.
It's excruciating. Every single day must feel like the clock is ticking down, and you are just sitting helplessly in the waiting room of your own life, hoping the cancer isn't spreading to your lymph nodes while some machine in another state processes your tissue.
Precisely. And while the patient is paralyzed by waiting, the tumor is not waiting.
No.
It is actively metabolizing, it is potential metastasizing, and it is interacting dynamically with the host's totally unique biology. But eventually, the sequencing report arrives.
Okay, so they finally get the results.
Right, and it basically generates a text file of mutations. The oncologist then takes those specific mutations, say like an EGFR mutation or a BRAF mutation, and matches them against a massive clinical database.
And Mishra calls this database a lookup table, which, I mean, from a computer science perspective, is literally just a basic reference array, right?
Exactly.
It's just input A equals output B. If the text file says mutation X, well, prescribe drug Y.
That is the mechanical reality of the lookup table. And here is the damning truth that Mishra exposes about this whole system. The doctor then prescribes the standard of care for patients with similar mutation profiles based entirely on population averages.
Wow.
They are not treating your cancer, they are treating cancers like yours.
Cancers like yours. That phrase just hits so hard because it totally exposes this massive, lethal blind spot in modern medicine.
It really does.
I mean, the source material emphasizes over and over that unique tumors have unique metabolic vulnerabilities, chemical quirks, and drug sensitivities that nobody ever bothers to look for.
Because the system just assumes that if the genetic code of your tumor is similar to the database, the behavior of your tumor will be identical to the database.
But that's not how biology works, is it?
Not at all. Biology is incredibly messy. It is not a simple, clean execution of software code. Two tumors with the exact same genetic mutation can behave radically differently.
Wait, really? Why?
Because the tumor's behavior is influenced by the patient's individual metabolism, the state of their immune system, the specific microenvironment of the surrounding tissue, even their diet or their stress levels.
Oh, wow.
None of those variables are captured in a genomic lookup table.
Okay, let me make sure I'm wrapping my head around the mechanics of this failure. It's kind of like taking a highly customized, heavily modified sports car to a mechanic because the engine keeps stalling out.
Okay, I like this.
Right. But instead of the mechanic popping the hood, turning on the engine, and physically listening to the misfire to see what's actually wrong, they just, type the VIN number into a computer.
Yes.
And they look up the factory manual for the base model sedan and just order the standard replacement part.
That analogy captures the diagnostic disconnect perfectly.
Because the factory manual might tell you where the spark plugs generally sit, but it completely ignores all the unique aftermarket quirks, the specific wear and tear, the custom tuning under the hood that are actually causing this specific car to stall.
Right. By relying purely on the manual, you are trying to fix the idea of the car, not the physical machine sitting in your garage.
Exactly.
And the consequences in oncology are just devastating. Because if that first factory manual fix doesn't work, if that targeted therapy fails, because your specific tumor had, say, a metabolic workaround, you have lost months of irreplaceable time.
Months you can't get back.
Exactly. You have endured severe toxicity from a drug that didn't even help, your immune system is now battered, and worse, the cancer has had time to adapt to the drug and potentially mutate into a more resistant form.
So, okay, if the genetic code matches the drug design, why does it actually fail at the cellular level? Why are we still using this base model manual if we know tumors adapt differently?
Because, as Mishra points out so clearly, modern biology has a fundamental measurement problem.
A measurement problem.
Right. We simply lack the tools to measure live behavior accurately at scale. And this measurement problem becomes a catastrophic bottleneck when we try to apply artificial intelligence to medicine.
Oh, and this is where the source material gets incredibly fascinating.
Yeah.
Because, everyone right now is talking about AI.
Constantly.
Every biotech startup promises that their, proprietary machine learning model is going to cure everything from Alzheimer's to leukemia.
Mm-hmm.
But Mishra takes a very contrarian, very grounded view on this.
He really does.
Yeah, she writes, biological foundation models are powerful, But models without measurement are just elegant speculation. They need ground truth.
Elegant speculation. It is such a brilliant distillation of the whole problem. Large language models and biological foundation models are magnificent at pattern recognition. But an AI is entirely constrained by the quality and the nature of the data it's trained on.
Right. Garbage in, garbage out.
Exactly. And currently, the data generated by biological systems is fundamentally forensic.
Okay, let's unpack that word, forensics. We usually associate that with, like, a crime scene investigation, CSI stuff.
And that is exactly how modern pathology operates. We rely on snapshots of the dead. Think about how a biopsy works. A surgeon cuts out a piece of your tumor. The very act of removing it cuts off its blood supply, fundamentally altering its state.
It's basically dying the second they cut it out.
Yes. Then, that tissue is dunked in formaldehyde to fix it, or encased in wax, sliced razor thin and stained with chemicals. Or, for genomics, it is ground up in a centrifuge to extract the DNA. We are freezing biological material in time, killing it to observe it.
So the data we are feeding these massive AI models isn't how a cancer cell lives. It's literally just a photograph of a dead cancer cell.
Exactly. From that dead snapshot, researchers and algorithms make inferences about what the living system was doing before it was killed.
That seems so backward.
It is. And then we introduce delayed feedback into the loop. The doctor gives the patient a highly toxic drug and then waits 3 to 6 months to see if the tumor shrinks on an MRI. Snapshots, inference, and massively delayed feedback. That is forensic biology.
And Mishra's demand in the face of this is so simple, but it completely flips the paradigm. She states, if it were my life, I would want film.
Not a photograph of the battlefield after the war is over, but a continuous, high-definition live video of the biological event exactly as it unfolds.
But wait, I have to jump in here with a genuine challenge because, if I've been following science news for the last 20 years, I've been told a very different story.
I know exactly what you're going to say.
Right. We mapped the human genome. We poured billions of dollars into understanding transcriptomes, mutation landscapes, CRISPR.
Yep.
So if my tumor has a mutation that explicitly programs it to grow fast, and the doctor gives me a targeted therapy designed to block that exact genetic pathway, why doesn't it work? The DNA code matches the drug. Are we really saying that decoding the genome just isn't enough?
It is a critical question, and honestly, the misconception surrounding it is exactly why the pharmaceutical industry experiences a 90% failure rate in clinical trials.
90%.
Yeah. Now, genomics is vital. Mishra herself states she would still sequence the genome and the expression state. But we have to understand the profound gap between genotype, the genetic code, and phenotype, the actual physical behavior of the organism.
Okay, genotype and phenotype.
Right. You have to understand the difference between the architectural blueprint and the physical building.
Walk me through the mechanics of that gap. How does the blueprint fail to match the building?
Well, genomics tells you what the code says. It dictates the potential of the cell. But it does not directly tell you what the living system is actively doing, especially when you introduce a chaotic perturbation like a chemical drug.
Okay, I'm following.
Imagine your tumor's DNA has the blueprint for a specific protein receptor that a drug targets. The sequence confirms it is there, but is that specific receptor actually active right now? Is the protein folded correctly? Or did a sudden shift in cellular temperature cause it to misfold, rendering the drug totally useless? Furthermore, is the lipid membrane surrounding the cell too dense, preventing the drug from physically reaching the receptor in the first place?
So the gene might be screaming, I'm here, but the physical reality of the cell is completely blocking the interaction.
Precisely. To use a macro analogy, you cannot understand the dynamics of a traffic jam by staring at a static map of the roads.
Oh, that's good.
The map tells you where the highway is, where the exits are, and the speed limit. That is your genome. But the map doesn't tell you that a truck overturned in the left lane 5 minutes ago, causing a massive gridlock.
Right. The map just says it's a 65-mile-per-hour zone.
Exactly. To understand the traffic, you have to watch the actual cars moving in real time.
You cannot see the traffic by looking at a map of the roads. That makes total sense. You can print out the entire genetic code of a tumor, but that data is entirely static. It doesn't tell you how a cell behaves when it is actively fighting for its life against a chemotherapy drug.
And without that behavioral real-time data, without the film, your multi-million-dollar artificial intelligence model simply does not have a ground truth.
It's just guessing.
It's just generating hypotheses based on how millions of other people's static maps looked. It is guessing.
So, okay, Demanding a live film of a cell sounds incredible in theory, but how is that physically possible?
It's not easy.
Right, because if you cut a cell out of a human body, it immediately starts dying, like we talked about. You can't just put it on a glass slide and expect it to behave normally while you blast it with chemicals.
No.
So how do you keep it alive long enough, and how do you actually film it at a molecular level? Which brings us to the actual hardware Mishra built, Precigenetics.
This is where the sheer engineering brilliance of the manifesto really shines. Mishra didn't just write a philosophy, she built a machine to capture the ground truth.
Hardware.
Right. Precigenetics is described in the source as a microfluidic system with integrated spectroscopy that captures real-time spectral fingerprints of living cells.
Okay, we definitely need to break down those mechanics for the listener. When we say microfluidics, we are talking about manipulating fluids at a scale so unimaginably small that basic physics start acting differently, right?
Exactly. We are talking about microscopic channels significantly thinner than a human hair etched into a chip.
Wow.
At this microscopic scale, fluids don't mix chaotically like water sloshing around in a bathtub. They move in what is called laminar flow.
Laminar flow, what does that mean visually?
They move in perfectly parallel, predictable layers, like perfectly stacked sheets of water. This allows engineers to flow liquids like a patient's own blood plasma, targeted nutrients, or specific experimental drugs over a cluster of living cancer cells with absolute, exquisite control.
So it's not a chaotic environment.
Exactly. The microfluidic environment mimics the sheer forces and pressures of the human body, keeping the extracted biopsy cells alive, happy, and metabolizing outside the host.
So the cells are kept alive in these tiny controlled rivers. But how do we film them? What is the spectroscopy component actually doing?
Spectroscopy, at its core, is the study of how light interacts with matter. Every single molecule in existence absorbs, reflects, and scatters light in totally unique ways based on its chemical bonds.
Okay.
When you shine a specific, highly calibrated wavelength of light, like a laser, onto a living cell sitting in that microfluidic channel, the molecules inside the cell vibrate, the light scatters back, and that scattered light creates a unique, complex barcode, a spectral fingerprint.
And the massive game-changing advantage here is that the machine is reading this barcode without ever touching or harming the cell.
That is the crucial innovation. There is no waiting for chemical labels to bind, there are no toxic fluorescent dyes, and most importantly, you are not killing the cells to examine them.
Right, they aren't soaked in formaldehyde.
Exactly. The cells are alive, they are breathing, they are consuming energy, they are actively reacting to their environment.
And the source material lists exactly what this system tracks live: lipid content, protein conformation, nucleic acid states, and metabolic shifts.
Yeah.
Help me visualize what that actually looks like in practice.
Okay, imagine looking at a computer screen hooked up to this microfluidic chip. You are flowing a specific drug over living cancer cells taken directly from a patient's biopsy. As the physical drug molecules hit the cell membrane, you watch the data stream shift instantly.
Oh, wow.
You are literally watching the lipids, the fats making up the cell wall begin to destabilize. You can see the proteins inside the cell folding or unfolding in response to the chemical stress. You can watch the cell's metabolism, how it converts sugar into energy, suddenly spike as it fights the drug, and then violently crash as the drug overwhelms it.
Just mesmerizing to think about. We are literally watching the molecular machinery of a disease jam up and break down in real time.
You aren't guessing if the drug worked based on an MRI showing a shrunken tumor 3 months later. You know unequivocally if the drug is working 5 minutes after you apply it because you can physically see the target cell's internal supply lines getting cut off.
Okay, that raises a massive logistical problem.
What's that?
Well, a biological cell is an incredibly noisy, chaotic environment, right? Millions of chemical reactions are happening every single second.
Oh yeah, very noisy.
So if this spectroscopy system is tracking the lipid shifts, the protein misfolding, the metabolic pathways all simultaneously, how on earth does a human researcher look at that massive fluctuating spectral barcode and not just completely drown in a sea of chaotic data noise? Like, how do you even know what you're looking at?
The short answer is a human researcher can't.
Right.
The human brain simply cannot process a real-time spectral fingerprint of thousands of simultaneous chemical shifts. And this is exactly why the artificial intelligence component isn't just a trendy buzzword here. It is the vital load-bearing architecture of the entire system.
Okay, so the AI is stepping in.
Right. The machine learning model acts as the ultimate filter, instantly translating that chaotic spectral noise into coherent biological signals.
So the AI is essentially the interpreter of the film.
Exactly. But before we get into how the AI interprets the data to, rewrite biology, we have to ask a deeply pragmatic question.
Which is
What liquids are we actually flowing over these cells? If you have this incredible live feed, what chemical weapons are you actively testing on the tumor?
right. Because Mishra makes a brutally realistic point here in the manifesto. If your life is actively on the line, you don't have time to wait for a pharmaceutical company to invent a new weapon.
No, you don't.
Her exact words are, I don't have 10 years for a de novo drug program. I will die if I stop.
And that's the harsh reality. The standard pharmaceutical pipeline for a completely new de novo compound is this agonizingly slow bureaucratic behemoth.
It takes forever.
You have discovery, preclinical animal models, phase 1 safety trials, phase 2 efficacy trials, phase 3 large-scale human trials. I mean, it takes a decade and typically costs over $1 billion to bring a single new molecule to market.
And if you're the patient,
If you have an aggressive stage 4 cancer today, that 10-year timeline is a guaranteed death sentence.
so out of sheer survival necessity, her focus shifts entirely away from inventing new drugs and moves toward drug repurposing.
Yes.
Which is such a fascinating, almost hacker-like approach to medicine. You basically take drugs that have already spent a decade passing FDA safety trials for one specific disease, and you use them to attack something completely different.
Exactly. She advocates using existing, incredibly cheap off-label drugs that standard oncologists would never in a million years associate with a specific genomic profile.
Right. The source specifically mentions a statin which millions of people take daily just to lower their cholesterol, a metformin analog, which is a dirt-cheap pill used to manage type 2 diabetes, and an old generic antifungal medication.
Yeah.
The idea is that one of these incredibly common everyday pills might completely hammer a specific tumor's metabolism just by the sheer luck of its chemical structure.
But importantly, it isn't entirely luck. It is based on a profound shift in how we understand cancer itself.
How so?
Well, for decades, we viewed cancer purely as a genetic disease. But cancer is equally a metabolic disease. Tumors require massive, unsustainable amounts of energy to grow so fast. They hijack cellular pathways, fermenting glucose in bizarre ways to fuel their expansion, something known as the Warburg effect.
Okay, so they're huge energy hogs.
Exactly. So sometimes a drug designed to lower cholesterol in the liver just so happens to perfectly blockade the exact metabolic pathway a specific lung tumor is relying on to survive.
But the absolute tragedy is nobody in the legacy system would ever know that, because clinical trials only ever test the cholesterol drug on people with high cholesterol.
Right.
They don't test it on lung cancer because it doesn't make genetic sense, according to the lookup table.
Exactly. The legacy system relies entirely on theoretical logic. But if you have Precigenetics, if you have the microfluidic film, you don't need theoretical logic. You don't have to guess.
Test it.
You can just empirically test the cholesterol drug on the living lung cancer cell and watch what happens. And Mishra details a specific execution method for this, centered on screening combinations.
Yeah, she discusses screening drug A alone, drug B alone, and then testing drug A and drug B together.
What she is hunting for in that real-time spectral feed is chemical synergy. And the cellular mechanics of synergy are fascinating.
How did that work?
Often, you will flow drug A, let's say the statin, over the cancer cell, and the spectral barcode shows nothing. The cancer just ignores it. Then you flow drug B, the antifungal, over the cell, and again, nothing happens.
Okay.
But when you mix A and B together, they create a catastrophic cascading failure inside the cell.
Wait, why does combining two useless drugs suddenly become lethal?
Because of multi-pathway interference. The statin might not kill the cell, but it might slightly weaken the density of the lipid membrane. It alters the cellular armor. Right, so the antifungal couldn't get inside before, but now, because the statin weakened the wall, the antifungal floods into the cell, travels to the nucleus, and completely shreds the cancer's DNA replication machinery.
That's insane. And because Mishra is using this real-time spectroscopy, she isn't waiting months to see if a tumor shrinks on an MRI.
No, she's watching it happen.
She is actively watching this synergy or antagonism, where drugs actually cancel each other out, unfold minute by minute. She takes the agonizing weeks of waiting for a genomic lookup table and turns it into days of what she brilliantly calls empirical observation native pharmacology on my cells.
Observation-aided pharmacology. That is the absolute crux of the biological revolution she is proposing. You are no longer relying on a statistical model of how a drug should work based on an average human.
Right.
You are watching exactly how a drug does work on this specific patient's actual tissue.
It makes me think of an analogy. Imagine you are trapped in a locked room, and water is rapidly pouring in through vents in the ceiling.
Okay, stressful.
Very. You need a tool to smash the lock and get out. The traditional pharmaceutical model is like sitting down at a drafting table as the water rises around your ankles, designing the perfect laser-guided lock-picking mechanism, sending the blueprints to a manufacturing facility, and waiting 10 years for it to be built.
Knowing full well you will drown in an hour.
Exactly. But Mishra's approach with microfluidics and drug repurposing, it's like frantically looking around the room, grabbing a heavy brass statuette, an old piece of wire, and a rusty nail, and just rapidly testing how combining them can smash the hinges or pick the lock right now.
Yes.
You don't care that the brass statuette was originally designed to be a paperweight. You just care that it is physically heavy enough to break the door.
It is the ultimate pragmatic hack for survival. You are bypassing the multi-billion-dollar, decade-long pipeline by brute forcing existing known safe compounds against a live enemy and observing the results instantly.
So we have the microfluidics, we have the live, non-destructive spectroscopy, we have the repurposed, off-the-shelf drugs, creating synergistic combinations. But this real-time screening isn't just a clever, desperate trick to save one life.
No, it goes way beyond that.
When this live behavioral data is fed back into artificial intelligence, it creates a self-improving loop that fundamentally changes the entire discipline.
This is where we move from a brilliant diagnostic tool to a true systemic technological revolution. Mishra outlines a very specific four-step loop that transforms biology from an art into an engineering discipline.
Let's walk through that loop. Step 1. Models generate hypotheses. So the massive AI foundation model looks at the patient's data and predicts, based on my training, I hypothesize that combining this cheap diabetes drug and this antifungal will starve this specific tumor.
Exactly. Then, step 2. Live measurement tests them. You physically take those two drugs, flow them over the patient's living cells inside the microfluidic chip, and record the real-time spectral response. You capture the actual ground truth.
Step 3. Measurement improves the models. The AI receives the live data feed. It sees the physical result of its own hypothesis. It sees where it was right, and crucially, it sees exactly where its prediction failed.
And finally, step 4. The models make the next screen smarter. Because the AI has now ingested empirical ground truth, it instantly updates its own weights and biases. Its next hypothesis for the next combination of drugs is vastly more accurate. The system learns exponentially.
And Mishra states this with such absolute conviction in the text. She says, that is not a feature. It is a new operating system for biology.
If we translate this into the broader landscape of computer science, what she's describing is a perfect data flywheel.
Let's unpack the mechanics of a flywheel for a second, because it's vital to understanding how this whole thing scales.
Sure. A flywheel is a heavy mechanical wheel that requires an enormous amount of initial energy to start spinning. But once it gets going, its own mass and momentum keep it spinning faster and faster with less and less external effort. In machine learning, a data flywheel occurs when your product gets better as more people use it, which naturally attracts more people, which generates even more data, which makes the product even better in an unstoppable, accelerating loop.
It's like having an autocorrect on your phone. Initially, it's pretty clumsy, but the more you type, the more it learns your specific vocabulary.
Yes.
But in Mishra's world, instead of learning that you actually meant to type ducking, the AI is learning the exact chemical password required to instantly shut down a lung cancer cell. And it gets exponentially faster at guessing that password every single time it runs a fluid test.
Precisely. But historically in biology, this flywheel has been fundamentally broken because the feedback loop, the ground truth, takes months or years to collect from human clinical trials.
Right, because of the forensic snapshots.
Exactly. By giving the AI an immediate physical ground truth within minutes or hours using the microfluidic chip, the AI stops guessing and starts knowing. It completely solves the hallucination problem that currently plagues biological modeling.
Because it's anchored in reality.
Exactly. An AI cannot hallucinate a cure if its homework is instantly graded against physical reality.
I really want to highlight the profound shift in terminology here. She doesn't call this a new clinical pathway or an innovative therapeutic protocol. She deliberately uses pure tech jargon. She calls it an operating system.
Yes.
The implication is massive. Calling it an operating system implies that the human body and its devastating diseases are finally being treated as a programmable, debuggable environment.
It implies a transition from observation to absolute mastery. You don't just passively observe an operating system, you command it, you rewrite the code, you debug the errors.
And the future trajectory she plots for this OS is staggering. She states that with the relentless work they are doing on tracking drug resistance, toxicity, and cellular targets, tomorrow they will be able to generate completely new compounds entirely in silico, meaning entirely via computer simulation, using AI, and just one piece of biopsy tissue.
This is the ultimate endgame of the biological data flywheel. Right now, she notes that the accuracy of their digital predictions against the physical reality in the chip is 97%.
Which is incredibly high.
And because of the flywheel, it will only climb. When that accuracy hits 99.99%, you no longer need the physical microfluidic chip to painstakingly test thousands of compounds.
Wait, really?
You can simulate the physical chip entirely inside the computer, test billions of novel chemical structures in seconds, and only ever physically synthesize the single, perfect, customized cure for that specific patient.
It is absolutely mind-bending. But we have to realize that this technological leap doesn't just save lives. It entirely shatters the current economic model of the global healthcare industry.
Oh, completely.
We have to spend time exploring the economics of time and money here because the contrast is staggering.
The financial implications are arguably just as revolutionary as the biological ones. We discussed the current standard of care, the genomic lookup table. Let's look at the financial reality of that system.
Right. The source notes that a single round of targeted cancer therapy, which, remember, is based on a generalized population average, and has a massive probability of failing against your specific tumor cost between $10,000 to $30,000 a month.
The system is asking a patient or their insurance to pay $30,000 a month to essentially act as a biological trial and error subject.
It's wild when you put it that way.
And as Mishra pointedly notes, the ultimate cost of a failed first-line treatment isn't just the crippling financial debt. It costs months of a patient's life.
Which is the one currency you can absolutely never get back. You pay $30,000 to lose 3 months to a drug that didn't work.
Yeah.
But then she outlines this profound financial epiphany regarding her own platform. She writes, "It cost us millions to develop our hardware, our infrastructure. But here's the funny part. Now that we already have all of this, how much would it cost extra to save my own life?"
This introduces a fundamental shift in how medicine is priced the concept of marginal cost.
Let's break that down.
The legacy healthcare industry operates on incredibly high marginal costs. Every single new pill manufactured, every new intravenous infusion, every hour a patient spends occupying a hospital bed costs massive amounts of physical resources and human labor. The cost scales linearly with the number of patients.
But Mishra's observation OS operates on digital economics.
Exactly.
Yes, the initial R&D, the prototyping of the microfluidic hardware, the massive cloud computing infrastructure, the software development, the sunk cost was millions of dollars. But to run one more patient's cells through the chip, to let the AI run one more simulation loop to find a 5-cent generic statin that will cure them.
It's nothing.
The marginal cost of doing that is basically just a few cents of electricity and a few drops of chemical reagents. The marginal cost is practically zero.
And returning to the flywheel effect, she accurately predicts that eventually this paradigm makes the cost of personalized treatment practically non-existent as the entire process moves in silico.
Right, when it's all simulated.
Once the AI perfectly maps human cellular response, discovering your personalized cure drops to the fractional cost of cloud computing.
I really want you, listening right now, to pause and consider the economic and moral absurdity of this contrast.
Well,
We are looking at a very near future where the true cost of perfectly simulating a personalized empirical cure for your specific cancer drops effectively to zero.
yeah.
On one side, you have a perfectly tailored cure found using a 5-cent repurposed diabetes pill. On the other side, you have a legacy healthcare industry demanding a $30,000 a month guessing game. When the technology scales, how on earth does the old system justify its existence?
It won't be able to. The economic collapse of the generalized pharmaceutical model is an inevitability in the face of an observation-native OS.
It's like Blockbuster and Netflix all over again, but for human lives.
It is entirely analogous to the collapse of the multi-billion-dollar physical encyclopedia industry when Wikipedia and algorithmic search engines democratized information retrieval. You cannot charge a premium for a guess when absolute certainty is virtually free.
It is an unstoppable technological force. But we have to ground this. Behind this massive technical framework, behind the complex fluid dynamics, the AI flywheel, and the impending economic disruption, there is a deeply human, honestly heartbreaking catalyst for all of this.
Yes, it's not just data.
This entire manifesto isn't just an intellectual exercise in computer science for Parmita Mishra.
No, the motivation is profoundly personal. We must look at the ghost in the machine, her explicit personal why that drives this relentless engineering.
She writes that she built this entire system because she watched someone she deeply loved, her aunt, die in a medical system that, in her words, could not see fast enough.
Her aunt was struck by ALS, amyotrophic lateral sclerosis. It is a devastating, relentless, progressive neurodegenerative disease. And as Mishra details the tragedy, her aunt didn't even know what disease she was fighting at first.
Because of the diagnostic delay?
Right. The diagnostic muddy waters we talked about at the very beginning of this deep dive. ALS is notoriously difficult to diagnose definitively until the damage is severe.
The helplessness she describes in the text is palpable and terrifying. Her aunt progressively lost the ability to move, to speak, and eventually to breathe.
Yeah.
She was locked inside her own failing biology, and the medical system was completely blind. It was too slow, too reliant on forensic snapshots to offer any real intervention to halt the cellular decay.
It is crucial to anchor all of our highly technical discussion about spectroscopy and AI in that raw emotional reality.
I have to.
The grand concept of an operating system for biology, the obsessive drive to etch microfluidic channels, to perfectly calibrate lasers, to program self-improving AI loops, it was all birthed directly from the acute, agonizing grief of being totally blind to what was happening inside a loved one's body as they faded away.
A system that could not see fast enough. That specific phrasing is going to stick with me long after this conversation ends. But out of that profound darkness, she has this realization of incredible, defiant empowerment.
It's a huge pivot.
She writes, "Today, I realized something incredible. If my own life were on the line, I, a biologist, know what I am building could actually save me."
It is a monumental transition from the trauma of helplessness to the absolute agency of the engineer. She realizes that the intricate tool she forged out of the grief of losing her aunt is exactly the tool she would demand to use to save herself. I would want to watch. That is exactly what I built.
I think it is such a beautiful, full-circle moment of human resilience. The deepest grief built the machine, and now that very machine is engineered to prevent anyone else from experiencing that grief.
It really is.
It is the ultimate testament to human ingenuity, taking the absolute trauma of biological helplessness and engineering it into the ultimate tool of empowerment. It transcends science. It is a literal reclamation of autonomy over our own flesh and blood.
And that profound sense of reclaiming autonomy, of radical technological empowerment, forces us to zoom out and look at the broader context of this document, because this monumental biotech breakthrough isn't happening in a vacuum.
Right, it's not isolated.
It is deeply embedded in a specific cultural and political moment.
It absolutely is, and we have to explore where this manifesto actually lives. Let's look at the backdrop of the world in March 2026, because the blog platform amplifying this breakthrough is intensely obsessed with a rapidly converging, chaotic, and technologically exploding world.
Yeah, let's talk about Bhagat's blog.
Let's unpack what the blog's author, Paramendra Kumar Bhagat, refers to as the impending age of abundance.
The framing Bhagat provides is fascinating, and it borders on the theological. He isn't just neutrally reporting on Mishra's biotech company.
No, not at all.
He is actively framing her engineering as concrete proof of a broader, almost prophetic shift in human history. He literally refers to her work as evidence of the impending age of abundance prophesied in the scriptures thousands of years ago.
Which is a wildly dramatic way to introduce a microfluidic AI chip.
Yeah.
But the conversation gets very real, very fast. Bhagat immediately transitions from prophecy to raw capitalism. He starts publicly tagging major venture capitalists on the post. He tags Peter Kolchinsky, Tony Kulesa, Life's IVC. He is openly demanding to know why Mishra hasn't raised gobbles of money already to scale this operation globally.
It perfectly highlights the hyperspeed at which information, narrative, and capital are expected to collide and mobilize in this era. And it isn't just niche venture capitalists taking notice.
Yeah, the big players are watching.
The source material shows that this highly technical biotech thread caught the attention of the absolute highest echelons of tech titans. We see a direct reply from Elon Musk to the thread.
Oh, wow.
Musk simply replies with 2 words: big difference, accompanied by a link.
Big difference. It is cryptic, sure, but it demonstrates that this core concept of transitioning to an observation-native OS has penetrated the awareness of the people actively building the future infrastructure of the planet.
Absolutely.
And to truly understand the chaotic zeitgeist of March 2026, we have to look at the incredible whiplash-inducing mix of topics surrounding this single post on Vagat's blog.
The blog's sidebars, categories, and reference book titles provide a vivid window into a world undergoing intense, multidisciplinary convergence. On the technological side, you have the sudden explosion of what is called agentic AI.
Stop right there. We went from curing cancer to agentic AI. How does a biological chip connect to autonomous artificial intelligence, and what actually is agentic AI?
Agentic AI represents a massive leap forward from the chatbots of the early 2020s. And agentic AI doesn't just generate text or answer questions, it is granted agency to execute complex, multi-step tasks in the real world autonomously.
So it does things on its own.
Exactly. It can browse the internet, write code, run experiments, and correct its own errors without human prompting. If you connect an agentic AI to Mishra's microfluidic chip, the AI isn't just analyzing data, it is autonomously deciding which chemical to flow next, running the physical fluidics, observing the result, and iterating entirely on its own.
So the AI becomes the actual scientist at the bench. That is staggering. And alongside agentic AI, the blog mentions concepts like liquid computing, references NVIDIA's CEO Jensen Huang alongside Elon Musk, and promotes an overarching framework called calciism.
While the source doesn't define the exact tenets of calciism, the context implies it is an ideological framework, a blend of macroeconomic and perhaps spiritual blueprints designed to navigate this specific age of abundance.
Where extreme technological capability theoretically eliminates traditional resource scarcity.
Exactly.
But, and we have to contextualize this completely impartially, just observing the data provided we have to note the intense, jarring contrast on this blog.
The geopolitics.
Yes, because right alongside this utopian vision of technological salvation and infinite abundance, the blog is absolutely steeped in the reality of intense global friction.
It's a stark contrast.
Just looking at the political tags from the exact same week as the cancer cure, rising Middle East tensions, Iran, Russia, Donald Trump's trade war and tariffs, AOC's 2028 political ambitions, and loud calls for a radically reorganized United Nations.
And to be clear, we aren't taking a stance on any of this.
Oh, no, absolutely not. We're just reporting the context of the blog.
Right. It is essential to recognize that this is a portrait of a world in deep, agonizing transition. As the blog notes, artificial intelligence, robotics, crypto, and synthetic biology are eating everything. They are rewriting the rules of reality.
Yeah.
Yet this explosion of capability is set against a backdrop of severe, entrenched geopolitical volatility. The old legacy systems of human governance, borders, and scarcity-based economics are clashing violently, even as the new systems of technological abundance are exponentially accelerating.
I love how perfectly this captures the human condition. Think about the pure dichotomy on that screen. You have human beings fighting over imaginary geopolitical lines, engaging in trade wars, and squabbling over legacy economics on the left side of the page.
Right.
And on the exact same screen on the right side, you have a lone biologist literally hacking the foundational code of organic life to cure our most devastating diseases using autonomous AI and repurposed 5-cent drugs.
It frames the ultimate race of the 21st century. It is the friction of human politics and legacy institutions racing against the relentless, compounding ingenuity of human technology.
It really is a race.
And in Parmita Mishra's work, we see a brilliant, blinding glimpse of what happens when ingenuity wins, when we stop blindly guessing and finally build the tools to observe reality as it is.
Okay, let's distill this incredible journey down, because we have covered massive ground today.
We really have.
We started in the agonizing, suffocating reality of the modern oncology waiting room, a system that forces a 3-week wait for an educated guess, resulting in a targeted therapy designed for a generalized population average.
Yeah.
The tragic era of treating cancers like yours.
And we explored the necessary paradigm shift away from that forensic, static, death-based model of genomics. The realization that you cannot understand the traffic by staring at a static map of the roads.
We moved from the map to the live traffic via Precigenetics, the engineering marvel of a real-time, live-streaming, non-destructive microfluidic operating system.
Right.
A system that flows cheap, repurposed, off-the-shelf drugs over your unique living cells and uses spectroscopy to watch the chemical battle unfold live, molecular shift by molecular shift.
A system that turns months of agonizing waiting into days of empirical observation-native pharmacology, and crucially, a system that feeds that physical ground truth back into an AI data flywheel.
The autocorrect for biology.
Yes, an autocorrect that will eventually make the cost of simulating a personalized cure drop to the near-zero cost of cloud computing.
So why does every single detail of this matter to you listening right now? It matters because the very philosophy of what medicine is fundamentally shifting beneath our feet.
It's a total rewrite.
Medicine is moving from a generalized statistical art form based on averages to a highly personalized, empirically driven, real-time engineering science. The era of treating cancers like yours is finally ending. The era of treating your exact cancer is beginning.
And it is vital to remember that this future is being built by people who understand the stakes intimately. It is driven by the profound, deeply human desire to never, ever have to watch someone they love suffer in the dark again.
It is an incredible triumph of the human spirit. But before we wrap up this deep dive, I want to leave you with one final, slightly provocative thought to mull over.
Okay, lay it on me.
Something that builds logically on everything we've talked about today, stretching the implications just a bit further. Let's explore the logical extreme of the technology.
I'm intrigued.
If Parmita Mishra and her team are successfully perfecting a closed-loop, AI-driven operating system to watch exactly how our cells react to any chemical in real time,
Right.
and if she can use that system to cure devastating disease by finding the perfect metabolic hack to shut down a tumor, how long until we use this exact same observation OS not just to cure the sick, but to actively upgrade the biology of the healthy?
You are pointing toward the inevitable shift from therapeutic intervention to active human enhancement.
Exactly. Think about the mechanics we just discussed. If we can map a tumor's metabolism in a matter of hours and hack it into submission using a combination of common statins and antifungals, what is stopping us from using that exact same microfluidic chip to map our own healthy cells?
Ethically, a lot, but technologically, nothing.
Technologically, to find the perfect chemical combinations to optimize our baseline cellular energy, to radically slow the aging process at the lipid level, to enhance cognitive function based on our own perfectly mapped real-time biological data.
It is the entirely logical, if highly controversial, next step of the biological data flywheel.
Oh.
Once you have built an operating system with read and write access to the human cell, you don't just use it to delete malware and viruses.
No.
You inevitably use it to write vastly superior software.
And that changes everything. We started this deep dive talking about the comforting illusion of the X-ray machine, the psychological safety of that jagged white line, the simple binary comfort of broken or not broken. We saw how terrifyingly murky the waters of complex cellular biology truly are. But maybe, just maybe, this new technological lens of real-time AI-driven observation isn't just going to clear up the muddy waters of disease. Maybe it is going to reveal an entirely new ocean of what it fundamentally means to be human.
A truly profound horizon to look toward.
Thank you for exploring the deep end with us today. Keep questioning the consensus, and we will see you next time.